Markus, please tell us about circular structures in high dimension gene expressed data.
Thank you. Thanks for the invitation and the kind introduction. Um, so I'm gonna talk a little bit about gene expression data, but it's not, the actual data set is not too important, and I want to focus more on the first aspect of the title, the circular structures in high dimensional data. But we're just using gene expression as a model here, and so for that, I'll just briefly explain the data set. And so this comes from a technology called single-cell RNA sequencing. And in its essence, it's a way to represent individual cells, cells from a tissue, for example, actual biological cells, into some kind of high-dimensional space, right? And the way it works is like we take this tissue and we dissociate the individual cells, and then for each cell, we measure its so-called gene expression. So this is, think of it as a positive real number for each gene. And so since the human genome consists of roughly 20,000 genes, we get for each cell can be represented in a 20,000 dimensional space. And this is how we end up. So usually we get this huge, massive matrix. And usually the way we think about it is in terms of a point cloud lying in some high dimensional space. And just to give you some numbers, how what a typical the typical size of this point cloud, we roughly get a few thousand cells. And as I mentioned before, if we're talking about human cells, we have 20,000 genes. So that would be around 5,000 cells in 20,000 dimensions. So if you've been dealing with data before, then you know this is quite, I don't know, annoying to work with because the number of dimensions is much higher than the number of points. So it's quite difficult to make meaningful statements. And in order to tackle this, we're going to look at lower dimensional projections, right? And we're going to be a bit smart with our projection into a lower dimensional space, because if we just project into some random dimensions, and the dimensions here again represent genes. So if we just project into some random genes, then we just get a random point cloud. But if we know a little bit about the gene set that we're projecting onto, then the point cloud kind of automatically starts getting some shape, and we can relate the shape back to biological properties. So in particular, there's something called cell markers. These are genes that are expressed only in specific cell types, like, for example, keratinocytes or fibroblasts. And so if we project down into cell type markers, then what we expect is that we get this natural clustering of the point cloud into the individual cell types. And so this is how it looks in reality. So this is not just some theoretical considerations, but if we have the right pipeline, then this is actually kind of what we see. But now we can think of other shapes too. We can think of a cell state transition. So a state like a cell has like some state, and then it moves to do something else, or like kind of changes its behavior. And this is we can think of it as a trajectory. And if we have a cell state marker here for the initial state and the cell state marker here for the end state, then we should get something like a linear trajectory. And again, this is something that we observe in real data. But the shape I'm most interested in, and the one I want to talk about today, is circular shapes. And you can think of it as arising from periodic processes within the cell. And so the stereotypical example of a periodic process is the cell cycle. And so by that, I just mean a process that happens over and over again. And so the cell cycle is just the property of a cell's dividing. So it divides over and over again. And you can, for example, decompose it into different phases. It's sometimes called G1, S, G2, and M. And during these different phases, there are different genes that are controlling these phases or driving these phases. And so we get kind of this bunch of periodic functions on the intrinsic time of the cell that is sometimes called pseudotime. And now the trajectory of these functions is traces out an actual cycle in this high dimensional space, right? So here I just took the trajectory of the gene expression of cyclin F. Sorry, it's not on there, but cyclin F exists and is somewhere here. And cyclin B, which is this one. And then this in 2 dimensions would be a cycle like this. And again, with real data, it looks like this, right? So I was quite excited about this. That the cell cycle is an actual topological cycle, but then you go to the biologists and they're like, oh, wow, that is very interesting. And so it would be good to kind of take this to the next level and generalize this and make something more out of this. So that's why we define quite a general class of cellular processes that here I want to call cyclic cellular processes, and that in some sense mimic the cell cycle in this, let's say, topological behavior. And here I don't want to go too much into the biology, but basically homeostasis is a steady state of a cell. And so if you perturb this, for example, if there are some pathogens like bacteria or viruses, then you perturb the state of the cell. It has to attack, it has to trigger an immune response and then returns back to homeostasis. And so this is something that is not periodic, but cyclic in the sense that you go through a transition of states and then end up in an initial state that we obtained before. And so there are similar examples in cancer drug development and in wound healing. But yeah, for the sake of time and because the audience, I guess, is more interested in the math, I'm going to skip over this. But just to tell you, there are a lot of these processes out there. Okay, and so what's cool about cyclic processes in general is that they come with an intrinsic time that is often called pseudotime. And think of it really as a map from your space into the S1, into the one-dimensional circle, right? And here, this pseudotime is quasi-given by these different phases, and we just kind of map all the cells on this circle. And so mathematically, there's actually something quite deep going on here. Namely, the way we find these cycles is by something called homology theory, or more specifically, persistent homology. And in our case, we can identify homology groups with cohomology groups. And then what's happening here is that, and this is very specific for the one-dimensional cohomology groups, is that they can be identified with maps into the S1, right? This is not the fundamental group. This is not S1 into X. This is really X into S1. And so this is sometimes called a Brown's representability theorem. And it is quite unique for one-dimensional cohomology. It doesn't work in 2 dimensions. But yeah, let's go back to pictures. So this project deals kind of with on the biological side is that we want to find these cyclic cellular processes and we want to decompose it, like understand the different phases of these processes. And the first part, we're doing this using persistent homology, and the second part, we do this using the Brown representability theorem, or what is often called, what's it called? Do I have it here? Circular coordinates, in analogy to linear coordinates of manifolds, right? So because a map from X, from your space to R is like a coordinate function on a manifold. But if you have it to S1, it's like a circular coordinate. All right. So mathematically, these are the goals that we are tackling. Given like a high dimensional point cloud, we're looking for projections into some lower dimensional space. so that the lower dimensional space has a pronounced circular structure, right? And here, in this example, I show, you can see that it can be that the higher dimensional object X does not have a cyclic structure, but only if you project it, it starts showing this cycles. Then we'd want to identify this inner clock using this circular coordinates. And then we can do some downstream analysis, like figuring out which genes are actually driving the cycle, how much they contribute, with which phase they're associated, and so on and so forth. All right, how do we find these subspaces? So we use something similar to what in bioinformatics is known as gene set enrichment analysis, but we kind of hijack this entire apparatus and we just call it circularity enrichment analysis. And at the core of it is a ring score. So we need some kind of score given a point cloud. We want to define a score that tells us how ring structured this point cloud is. One being it's like a perfect circle or a perfect annulus, and 0 means it's, I don't know, maybe tree structured or something like this. Like absolutely not a ring, right? And then we just so we start with our molecular profile, which is our single cell RNA sequencing experiment, and then we just screen through a predefined collection of gene sets, which are just a predefined, think of it as a predefined set of projections. So we just, just need to screen through these projections, see if they have a high ring score, and then fish out the ones that have a high ring score. Okay, sorry. Here's what I said in words. Yeah, so we define a ring score. We use it to, the ring score has this property that it fishes out point clouds that are ring structured, and we screen through a database. All right, so how is the ring score defined? For that, we need, we use something called persistent homology. I hope most of you are familiar with normal homology, or at least have an intuition for that. Normal homology measures holes in some sense. And so persistent homology is doing this for data. And one way you can do this is with this construction. You start with your point cloud, and then you enclose a disk or a ball, a higher dimensional ball, around each of these points in the point cloud. And then you take the union of all of these balls, and this gives you an actual, like, interesting topological space. Because the problem is, if you start with the point cloud, then you just get a discrete topological space, so nothing interesting is happening here. If you take the union of balls, then something interesting can happen. And then you take the homology groups of this space at this given radius of the balls, right? And then what you can do now is you can think of an evolution of topological spaces. It's often called a filtration. So you just let these balls grow, and you keep track of topological features. In this case, we keep track of one-dimensional homology groups. And then we summarize this in a diagram here called a persistent diagram. So here's the time of birth of a topological feature, in this case, a one-dimensional homology group. And here the death of the feature. Yeah, I only talk about the green points now, the one-dimensional homology groups. So you can see, for example, since my space here mainly has two holes, like if you look at here, it has two holes, right? There are two persistent homology groups, whereas the other homology groups that emerge are kind of like noise, topological noise, in the sense that here they can microscopically or in small scale, they can just out of pure noise, small holes emerge, and then they die fairly fast in this evolution. And so this persistent diagram can tell us something about the global topological structure of our point cloud. And here in particular, we would say, okay, we have two persistent one-dimensional homology groups. And so, yeah, this persistent diagram is a structure that people usually work with. In this work, we introduce something we want to call persistent sequence, because we don't, there's a lot of information here that we don't really need. So we just simplify this, and we take the persistence, like the lifespan of each homology group, and we sort them by order, and we just keep track of the length or the lifespan of each homology group. So the lifespan is just time of death minus time of birth. All right, so we have this persistent sequence, and then we define the ring score out of this by this formula. Before you look in this formula, I'll just give you the intuition of how we designed it. So if something has, assuming there's no noise in your data and you just have a perfect circle, then the ring score is going to be 1, or perfect topological circle. Perfect, I don't know, potato. Then you have a ring score of 1. If you have 2 holes, then you have a ring score of a half. And in general, the ring score would exponentially decay in the number of holes, right? So the more spongy something is, the less ring structured it is. And we concretely, we kind of we take this philosophy that the most persistent homology group is the ring that we're trying to detect, and everything else is considered noise, right? So we just measure how far the noise is from the actual signal that we're trying to measure. And then we just sum it up and give it a weighting so that we have everything neat and nice between 0 and 1. There are some kind of caveats we have to be aware of, and that's namely that in some sense, topological transitions are discontinuous. So again, if you assume no noise and you have like a more eccentric, like a circle that becomes increasingly more eccentric, then the transition from very eccentric ellipse to a straight line is discontinuous topologically, in the sense that this has a one-dimensional homology group and this has 0. So, well, it's just I guess it's a direct consequence of using algebra to describe something continuous. You have to jump eventually if you use the discrete descriptors. But yeah, so we solve this. Yeah, this is particularly bad if you use noisy data, because you can have weird things at very eccentric yeah, ellipses. So yeah, that's why we normalize to the diameter, and we introduce kind of a new normalization, and that kind of solves the problem. Yeah, we have different variations of this, but the one you should keep in mind is just we normalize everything through the diameter so that this transition becomes continuous. All right, let's move on to the intrinsic clock. So we said if we have a set of periodic functions, one can plot the trajectory, and it will end up in a circle in a high dimensional space. And now what we want is actually the other way around, because we don't have this, but we want this. This is the thing that gives us biology. This is the thing that we measure. So I wanted to include more slides about this, but I kind of I'm still struggling to understand exactly how this works. So I'll just give some references. But yeah, this circular coordinates, this is something people have been working on since 2009. And roughly we use this Brown representability theorem. This one. And this still doesn't give you a function, or it doesn't give you a concrete function from your space to S1, but it gives you a class of homotopy equivalent functions. And the question is, how do you choose one that makes sense? And it turns out there is one unique representative, or one canonical one, that comes out from what they call a harmonic representative of uh, homology classes or cohomology classes. I don't wanna, yeah, it's if you heard about it, there's something called the uh, Hodge Laplacian on chain complexes, and it's the kernel of the Hodge Laplacian. All right. I guess what I'm saying here is there's a way to once you have a unique homology class that you're interested in, first to turn it into cohomology, since we're working in Rn and we have a scalar product and everything, and then from cohomology go to a function or a class of functions from your space to S1, and then you can smoothen out this function by choosing the harmonic representative. All right. This is just kind of a proof of concept that what we're doing kind of works in some sense. Here we have a data set of hematopoietic stem cells. These are blood cells, bone marrow cells of mice, and they're particularly useful because not every cell is actively participating in the cell cycle, but these ones do. And so we run our database of gene sets, and we fish out the gene sets that have a high ring structure. And again, think of the point cloud. We find the projections that where the point cloud projects into some ring structure, and we find the cell cycle, which is nice because it validates kind of it validates the biology or what we would expect from a biological point of view. And then we infer this pseudo time, which is the circular coordinate, and we can picture these things in 3 dimensions using principal component analysis, and we see that the circular coordinate really traces around the visual cycle, let's say. Then one thing we could ask biologically is if the topological cycle that we see is just the cell cycle, but kind of like weirdly projected into a different space. And if we assign the cell cycle phases, then we see that the cell cycle phases are nicely in line for the cell cycle pseudotime. We see a little bit of clustering here in the apoptosis one, but this interferon alpha response, we really think it's a different cycle other than the cell cycle. And then so the next step is to kind of go deeper into the biology and use more tools to really study this cycle. And so this is a summary slide. What we are doing. We are trying to define the concept of cyclic cellular processes in single-cell RNA sequencing data using topological data analysis. We developed a ring score to measure ring structures of these processes, or in general, ring structures of point clouds. And we established a pipeline to perform the circularity enrichment analysis to fish out the cyclic cellular processes. And we showed that it works in synthetic and in real-life data. And there's still some work to do to actually make the biologists happy. With that, I want to thank everyone who has been with me on this journey, and I want to thank you, the crowd, for listening.
Thank you, Markus. Let's just thank Markus for his talk. Okay, I believe we have a question from Bruno in the chat.
Yes.
What kind of groups do you get from the persistent homology? Is Bruno's question.
Yeah. So I assume you mean which coefficients am I taking? So computationally, we usually take Z2 coefficients, but the underlying assumption is that torsion doesn't really appear in real data. So ideally, you would take Z coefficients, like integer coefficients. but this is computationally quite expensive. So that's why we take Z2 and we just assume you can go from Z2 to Z. And this assumption is true if there is no torsion. And this is kind of an assumption about data in general. Does that answer your question?
Okay, sounds like it's all okay. So I think I think I've been to some talks about where just topological data analysis might have just been mentioned, and it was mentioned that it was very computationally expensive. Do you have any idea of how computationally expensive it is? Like, is it
Yeah.
exponential time or?
So the official complexity class is n to the 3, but in practice it's faster.
N to the 3, where n is the number of points, or?
Yeah, n is the number of points. And I don't think the dimension really, it's probably linear in the dimension, but it really doesn't make a difference, like the number of dimensions. It's really on the number of points that is more cumbersome or troublesome.
Mhm.
I think when people say that persistent homology is expensive, is that the higher homology groups are expensive to compute. So H1 is not so expensive to compute.
Thanks. Yeah, it's a good comment. Yeah.
Are there any more questions for Marcus?
I was wondering this d, the diameter, is that the diameter of the point cloud or what is that?
Yeah. Yeah, it's the diameter of the point cloud. And then this normalization comes from having that the perfect geometric circle has a score of 1. Right? Because if you have a perfect circle, then the circle, the hole closes when the radii of the points on the circle have a are if they are square root of 3/2. If you can inscribe this this triangle in the circle. But yeah, D is the diameter of the point cloud.
Yeah, thanks.
Are there any more questions? Okay, if there are no more questions, let's thank the speaker one more time.